Time to start today's presentation. Hi everyone, my name is Chang. I'm the host of today's presentation. Today we're going to introduce something very exciting and fancy. So it is about the biohybrid robotic. So it's a kind of new concept with half animal and half microelectronics. So as you see in the title, it says biohybrid robotic jellyfish using the microelectronics to drive the live jellyfish swimming. Today's presenter is Dr. Nicole W. I know Dr. Nicole W about maybe 2 years ago. I first saw her presentation in APS, which really impressed me. And right now, Dr. Nicole W is a bioengineer with an interdisciplinary background in robotics, fluid mechanics, and organismal biology. Currently, her research focuses on using a 3D printed biomimic shark spinning ropes to improve the performance of fish-like robots and autonomous underwater vehicles as a National Research Council postdoc associate at the U.S. Naval Research Laboratory. In her PhD work at Stanford University, Dr. Xu developed a robotic system to control the live jellyfish swimming, which has received international press, including the interviews on BBC Radio and TV. She received her MS in bioengineering from the California Institute of Technology and the BSc in the bioengineering from the University of Pennsylvania. So before I start officially welcome Dr. Xu to present her work, I'd like to remind you, if you had any question, please do not interrupt her presentation. You can wait and her finish her presentation, then ask a question in the Q&A session. So the total presentation will be about 45 minutes. So, and now let's welcome Dr. Xu to present her work.
Thank you so much, Chang, for the kind introduction. As you mentioned, my name is Nicole W, and currently I'm an NRC postdoc at the Naval Research Lab. But today I'll be discussing work that I conducted in my PhD with Professor John DeBerry at Caltech and Stanford, as well as some of our scientific collaborators at the Marine Biological Lab in Woods Hole, Massachusetts. So the motivation behind this work is to expand our capabilities in ocean monitoring. So we know that the ocean regulates the climate, helps modulate oxygen and carbon dioxide exchange, and provide food, medicine, and a source of transportation for both humans and animals. But we literally know a drop in the ocean. So over 80% of our ocean is still unmapped, unobserved, and unexplored. And it's important to learn more about the ocean so that we can track changes in temperature, acidity, and concentrations of nutrients so that we can detect and prevent situations like coral bleaching, as shown in the top between this healthy, beautiful, brightly colored coral versus a sadder, not brightly colored bleached coral, or harmful algal blooms like the one shown here in Lake Erie. It's also important to monitor the ocean so that we can discover new species and behaviors like this cute nudibranch or sea bunny, as well as to survey different areas over time. So the current methods of ocean monitoring include using remotely operated vehicles, or ROVs, and autonomous underwater vehicles, or AUVs. And these have a lot of great advantages. So they can travel very fast speeds over long distances and be used to collect data like ocean mapping or deep sea exploration. However, they can be really costly, they can be bulky, they can consume a lot of power, and leave larger vehicle wakes that have more noisy signatures in the ocean. And so our goal is really to use technology to collect data to learn more about new ocean environments, places that we haven't really reached before. And so to do that, we need new tools that are low power, low cost, and will minimally disturb aquatic life. So we can achieve this by combining various technological advances as well as experimental work and computational tools. And I'll be touching upon all three of those concepts today. One approach to doing this is to use bioinspired aquatic vehicles that can leave more natural wakes to reduce these environmental disturbances. So two examples I've shown here are the Aqua AUV on the left, which is a paddling swimming robot, and the soft robotic fish on the right. And these have both been deployed in real ocean environments before. And they've been used to collect images, take data, or look at samples from new locations. And so there's this question of what kind of model organism should we base these bioinspired robots on? And each model organism has its own advantages and disadvantages. So I've listed three different organisms here. One is the robotic fish, one is a manta ray, and one is a jellyfish. And so fish have advantages in terms of faster speeds and more advanced turning behaviors. Manta rays are flexible and agile, but in particular, jellyfish are very energy efficient. They're ubiquitous, meaning they're found in a variety of different environments, and they don't have a swim bladder, so they can traverse down to very deep parts of the ocean. And so, of course, to address the question, which animal model do we choose? The title of my talk and everything you've heard about me has already given it away, but today I'll be talking about jellyfish. And so hopefully I'll convince you that jellyfish are a great model organism to use, because despite over 500 million years of evolutionary pressure, their body structure has remained largely unchanged. So Aurelia aurita, or moon jellyfish, have a very simple body structure as shown here. Their swimming is intimately tied into behaviors like feeding, escaping, reproducing, etc. And they're found in a variety of different environments, including extreme temperatures, salinities, and depths down thousands of meters below the surface in the Marianas Trench. They're also invertebrates, and they don't have any pain receptors, which simplifies the ethical considerations of our work. And so one hypothesis as to why jellyfish haven't changed over all of these years is because they are very energy efficient. So we can look at the cost of transport as one metric of energy efficiency. And the cost of transport is defined as the mass-specific energy per distance traveled. And you can think of that as the inverse of miles per gallon. So a low cost of transport equates to a high energy efficiency. And so then we can look at a plot of the cost of transport on the y-axis over different masses of animals. I've also highlighted where AUVs also exist on this plot. And you can see that for a variety of different animals, so flyers, runners, as well as swimmers, swimming animals tend to have lower cost of transports in general. And I just want to highlight that this graph, that these axes are logarithmic. And so compared to all of these animals, jellyfish have the lowest cost of transport, which means that we might learn how to make more energy efficient robots by studying how jellyfish swim. But you can imagine that there is a huge range of how we can approach these bioinspired jellyfish constructs. So on one end of the spectrum is more of a robotic construct, as shown here with RoboJelly. And this is made using entirely engineered materials. And there are a lot of advantages, including well-controlled inputs and outputs, because you're able to control this robot entirely. But the limitations here are that this robot still consumes a lot of power. So you can see in the background, it's still wired to an external power source. On the other end of the spectrum is more of a biological construct, like this artificial jellyfish or medusoid made from seeding rat cardiomyocytes onto a silicon scaffold. And this is really great bioengineering work. It's a wonderful proof of concept that we can use muscles as actuators, which is ironic because I usually describe actuators as robotic muscles. But the limitation here is that these constructs can only survive in a few specific environments in the laboratory. So we can't send these out into the ocean right now. And so the approach that I'll talk about today is to use a biohybrid robot by combining a microelectronic system onto live animals. And this is advantageous because it's low power and low cost. We only need to wire this small microelectronic system. And it's also adaptable to a variety of environments because that's where these jellyfish naturally live. So jumping ahead to the very end of the story, this is just one example video that I'll also show you later on, but this is our biohybrid robotic jellyfish with a microelectronic system attached, and it's swimming upwards in the ocean. So today, I'll first be talking about how jellyfish naturally swim so we have a better understanding of its own natural behavior. And then I'll go into characterizing the artificial control of jellyfish muscle contractions in a series of tethered experiments in the lab. I'll then describe the process of building a portable swim controller, experiments using this swim controller in a biohybrid robotic jellyfish in the laboratory, as well as testing the system in real ocean environments in Woods Hole. And finally, I'll conclude by talking about improving robotic controllability for ocean monitoring in future work. So first, we need to understand how jellyfish naturally swim. And so this is an image of a jellyfish shown upside down. The top of the jellyfish is called the X umbrella, and the bottom of the jellyfish is called the sub-umbrella. On the right is an image of a jellyfish with the sub-umbrella surface facing us. So you can see along the edge of the jellyfish, in each of these 8 little circles is a pacemaker, which is a sensory organ that innervates the swim muscle. And the swim muscle is this coronal muscle that's 1 cell layer thick. And it's activated in an all or nothing pattern, which basically means that even if only one or a few of these pacemakers fires, each of these activated pacemakers will cause a bidirectional muscle wave propagation that eventually causes this entire muscle ring to contract. And so in order to swim, jellyfish have two different phases. The first is the power stroke, which is when the muscle actively contracts, and this provides motive force. So you can see that's the white section of this cumulative distance over time graph. There's also the recovery stroke when the animal is more passive and relaxed. And so you can see that about a quarter of the total distance traveled occurs in this yellow phase, which is the recovery stroke. So now that we have a basic idea of how jellyfish naturally swim, we're not limited by the jellyfish's own biology, and instead we can use electrodes to act as artificial pacemakers to directly activate the muscles ourselves. So this is a signal showing a square pulse wave of the voltage over time, and we can send these signals via electrodes to jellyfish at various locations. So we can change the different inputs by varying the voltage, the pulse width, the signal period or frequency, as well as the location of the bell as illustrated in these 4 X's here. And today I'll be focusing on the frequency as well as the location of the electrodes, but the outputs that we get are muscle pulses or contractions, and we're using the displacement of a red tag that I've injected into the jellyfish as a proxy. So I'll show you what I mean. So this is again a jellyfish shown a subumbellar surface facing us, and it's placed on a black dish. I'm going to highlight the bell just because there's lots of reflections from the light, so it's easier to see this way. And if you take a look at any of these 8 circles, you can see that I've injected this red tag. And so every time the jellyfish contracts, you can see a small displacement in that red tag. So using videos like this, we can then get a plot of the tag displacement over time. So this is just one example for natural animal swimming. So it's in the absence of any external stimulation. So we can see what the natural tag displacement over time curve looks like versus when we externally stimulate the animal at a quarter hertz. And so you can see a very regular pulse. These vertical black lines are when we are sending a signal to the muscle. And so you can see a very clear pattern following our stimulation. Sometimes there are double pulses. These are still animals that do their own thing. And so we can't necessarily arrest natural animal behavior, but it does follow our signal pretty well. It also follows our signal at 1 hertz, but once we go to frequencies that are too fast, the animal can't really keep up. And so there is a sweet spot for electrical stimulation. And we can successfully stimulate the animal at frequencies between a quarter hertz and 1 hertz very consistently. But instead of looking at the time domain, we can also look at the frequency domain. So this is one example of the tag displacement, but we can take 10 different animals and look at the mean amplitude over frequency. So for natural swimming behaviors, we see that natural motion includes this dominant frequency with a peak at 0.16 hertz, as well as this larger spread between 0.4 to 1 hertz. So that is what the animals naturally do. But when we stimulate the animals, so this is for an input of 1 hertz, we get a very different amplitude spectrum. So you can see that there's a very sharp peak at 1 hertz. So for an input of 1 hertz, we're getting a very clear output of 1 hertz. And so you can compare the natural versus externally stimulated spectra here. And we can take various power spectral density graphs for each specific frequency and then fold them into a plot of the frequency output over the frequency input. So this is a map in which each of these individual PSDs gets converted into one vertical line of data, and that's these white lines here. The data is interpolated, which is why it looks very discrete. But in theory, if we stimulated the animals at all the different frequencies, we should see a very smooth curve here. And so this amplitude, so this colored map is colored by the amplitude. So these higher peaks should be yellow, which is what you see here. And I just want to highlight a couple of things. So this red diagonal line is the successful one-to-one response. It's what we want to see. So for every input, the animal is giving us an output. There's also a red dashed line at the very top, which is the maximum pulse frequency according to a proposed biological limit from a previous paper. You can see that our animals can pulse slightly faster than that biological limit. And this thicker red dashed line at the bottom is the peak frequency of the unstimulated animals. So that's what the animals naturally do. And you can see that this graph basically gives us the same information as before, that we can see successful frequency responses between 1/4 hertz and 1 hertz. But I just want to highlight what's going on in this region when we're stimulating the animal above what it can do. So each of these cases can be split into responsive animals versus unresponsive animals. And you can see that this really depends on kind of individual animal responses. So there are some animal responses that can follow higher frequencies pretty well, whereas others really can't. So for future robotic control of these animals, we can then do some simple experiments in which we see, does this animal follow the frequency at higher frequencies? And then use those animals for robotic control if that's something that we're interested in. So another question that we had is, where is the optimal location to insert the electrodes onto the animal? So I tested 4 different locations as shown here, and we're really addressing the question, are electrodes at all locations embedded in the coronal swim muscle that I showed before? So to address that, I just want to take one step back first and talk about the life cycle of jellyfish. So until this point, I've just been speaking about adult jellyfish, but they have a very complicated life cycle that I won't get into. But you can essentially think of a phyra as baby jellyfish. So these adult jellyfish can be as small as 1 centimeter in diameter. So this is a little adult jellyfish. It's not a baby, even though it's very small. And the actual baby jellyfish, a phyra, are shown here. And they range between 1 millimeter to 1 centimeter in diameter. They kind of look like little umbrellas. So the reason I'm talking about this is that the muscle structure in younger jellyfish looks very different, or there's a question of whether it looks different between younger jellyfish and adult jellyfish. So this is a muscle stain in which one of my colleagues at Caltech stained the F-actin in jellyfish. And so you can see a very clear divide between the animal's coronal muscle versus radial muscles, as shown here. This is also true in smaller adult jellyfish. So this is a medusa that is 1 centimeter in diameter. And you can also see a clear divide between this coronal muscle and the radial muscle. So there's this question of how does the orientation of muscles change as the animals grow and age? Because the size of the jellyfish that I'm using for robotic control is about the size of a dinner plate. We also want to know how far inward does this coronal muscle extend. So when we place the electrode into the animal, is it touching this coronal muscle or the radial muscle, and does it matter? So to address that, I also did a series of F-actin stains in which I took a strip of tissue from a large jellyfish. I then stained it and then took images all along the center line to see how the muscle changed from the center of the animal to the margin. So you can see that closer to the center of the animal, we have these kind of wavy striations that then start to get more and more circumferential as we go outwards. And we can see that to address these 2 questions, how does the orientation of the muscles change as the animals grow and age? There's not really a sudden transition point. This is all very smooth. And this coronal muscle is extended basically the entire subumbellar surface. So the electrodes are placed at all, on this coronal muscle ring at all locations. And so we would expect to see very similar frequency spectra at all 4 locations. And that's exactly what we see, that the frequency spectra looks very similar. And so we can place the electrodes anywhere for consistent muscle responses on this jellyfish. So now that we have an idea of what electrical signals can successfully stimulate animal muscle contractions, we can then build a portable version, so a swim controller. And there were a few different design choices that I considered. We wanted something small, neutrally buoyant, so it's not pulling the animal down or upwards, and something that is low power and inexpensive. So here are the components that I used, including this housing, which is made from a plastic cap, as well as a bit that embeds into the animal. There is a piece of plastic on the top just to waterproof everything, a microcontroller, a battery, as well as 2 electrodes that flash red every time there's a signal being sent, just as a visual confirmation. And so this is what the swim controller looks like once it's all put together, but without being embedded in the animal. So you can see in the very front is an inactive swim controller, but in the back, you can see that the 2 electrodes are flashing red. And this is what it looks like once it's embedded into the actual animal. And this is what it looks like when it's embedded in the actual animal in the ocean. So here is just a cartoon schematic so that you can see some of the components as I talk through them. But I also just want to highlight that because these animals are invertebrates and they don't have any pain receptors or a brain, this simplifies the ethical considerations, but I have thought in more detail about the ethics of these experiments. So you can go to this QR code above if you want to hear more about the ethics of biohybrid robots in general, as well as using our biohybrid robotic jellyfish as a specific example. So this is a video of this biohybrid robotic jellyfish. I'm going to put up the QR code for the ethics paper again as we talk through it, but you can see that our biohybrid robotic jellyfish looks very different from the other natural jellyfish. So this is an environment in the lab called a pseudochrysal, which is a circular flow tank, and you can see that there's a background flow. In fact, you can see all the detritus and natural particles that are flowing in the water. And if you look in the background, you can see that the jellyfish that don't have a swim controller are swimming very passively. They don't swim as regularly, and they're more carried by the current in this tank, as opposed to the biohybrid robotic jellyfish that swims downward very strongly at a regular pulse. So now I want to talk about a series of experiments that we did in the lab to figure out how swimming frequency affects swimming speeds. So this is a series of experiments in which we had a jellyfish with 2 electrodes for symmetric straight swimming. We had the animal swimming downwards in a 2-meter tall tank at various frequencies. And these are some example videos of what these experiments look like. So we had a control in which the swim controller is embedded but inactive, as well as when the swim controller is pulsing at 0.5 hertz and at 0.88 hertz. So you can see that the kinematics actually changes. At 0.5 hertz, the animal is able to contract and relax fully, whereas at too high of a frequency, it's kind of curled up and it's unable to fully relax to provide as much thrust. And from videos like this, we can then look at the enhancement factor over the swim controller frequency for a variety of different animals. So this is one animal with a fineness ratio of 0.3, and the fineness ratio is defined as the height over the diameter. So you can see that for this animal, it can swim up to almost 3 times faster than what it would naturally do. But for an animal that is more oblate, that means it's flatter, then we're only seeing a very modest increase in swimming speed. And so for a range of animals, we tested 6 different animals of various fineness ratios. We can see a very clear pattern here. So we can increase jellyfish swimming speeds up to 2.8 times what they would naturally do, but this enhancement really depends on the body morphology or the shape of the overall animal. So these rounder, more prolate animals can swim faster than the flatter or more oblate animals. We also wanted to test what this would look like in a mechanistic model. So this is a theoretical model that incorporates the body kinematics of the jellyfish in terms of thrust, drag, and acceleration reaction forces. And we're starting from Newton's second law. So you can see the components here: thrust, drag, and acceleration reaction. And we're balancing the forces to obtain swimming speeds. So the input is information about the animal, including the kinematics, and the output are the swimming speeds that we get. So we can get a very similar curve in which we plot the swimming speeds over a variety of different frequencies. And we also see a similar pattern in both our theoretical and experimental jellyfish, that the rounder, more prolate animals tend to swim faster than the flatter, more oblate animals. And this is important because for future robotic control, again, we can look at how round a jellyfish is and choose those rounder animals to have faster swimming speeds in the ocean. So it's great that we can increase jellyfish swimming speeds, but there's this question of what energetic cost is this to the animal, because the animal is still consuming a lot of energy in these experiments. So to test this, I conducted a series of experiments in which we had an oxygen probe that was both in the water as well as embedded into the animal tissue itself, so that we could get bulk measurements of the oxygen and the tissue. And so from experiments like this, we were able to plot the oxygen concentration depleting over time. And this is one example. And we can combine all the data into oxygen consumption rates for animals at different swimming frequencies. So you can also see that when the animals are swimming at faster frequencies, they are consuming more oxygen, but once they get to too high of a frequency, they're not actually consuming as much oxygen as at lower frequencies. So we can take that data and fold that back into the cost of transport that I mentioned at the beginning of this talk. So this plot shows the cost of transport over various swim controller frequencies. And this blue point is the cost of transport for moon jellyfish naturally found in the literature. And so our baseline actually falls within this, which is really great. We also calculated the cost of transport for two different models. One is a power law using the swimming speed of the animal, and one is that mechanistic law that I showed you before with that hydrodynamic equation. And using our experimental data, our cost of transports are shown here in black. And so I just want to highlight that at this particular point, we're able to enhance swimming speeds up to almost 3 times what they would naturally do, with only a 2 times increase in the cost of transport. And so this suggests that there is a mode for both faster and more energy efficient swimming in jellyfish. So it's great that we can calculate how much energy the animal is actually consuming, but there's also this question of how much power the robotic system is consuming. So we can look at the microelectronics and compare this to various swimming robots in literature. So this plot shows the external power per mass for a variety of different robots over swimming speeds. And the marker shape accounts for the different type of robots, so more biological robots, so using rat cardiomyocytes, over more mechanical traditional robots. And the color corresponds to the type of propulsion. So I just want to highlight a couple of different robots. The first is the Jellyfish, which is this soft robotic jellyfish created by a scientist named Jennifer. So I really love that pun. There's also the robotic stingray shown here, and that's created using those rat heart cells. And the Remus 100, which is an AUV. So compared to all of these different swimming robots, we can see that our biohybrid robotic jellyfish exists here. So we're using between 10 to 1,000 times less external power per mass compared to other UUVs. And so there's also this question of how this system would perform in the ocean, because our ultimate goal is to take these biohybrid robotic jellyfish and have them swimming in the ocean and collect data for us. But of course, the ocean is a very different environment from laboratory experiments. So to figure out how the animals would last in non-quiescent and natural environments, my collaborators and I went to the Marine Biological Laboratory in Woods Hole, Massachusetts. And here are the names of our scientific scuba divers here. This is a map of Massachusetts. And if we zoom in on this red star, we can see where Woods Hole is. And if we zoom in on Woods Hole, you can see some images of my collaborators here. They're the scientific scuba divers in the water. Here I am on shore, completely dry the entire time. In fact, I asked a passerby to take a photo of me so that I had some proof that I was there. And here's a styrofoam box with some of our jellyfish and extra swim controllers, because nothing ever goes right when we're doing field tests, right? So we wanted to recreate the results that we got in the laboratory. So you can see this is an example of what the laboratory experiments look like. And this is what the field experiments look like in a cartoon schematic. So we had the animals with this 2 electrode swim controller, and the animals swam to the very top. We had one scientific scuba diver with a camera imaging the animal. And in the background is this rope with alternating red and yellow markers so that we could get a ground truth. So where's the jellyfish relative to the markers in the background? And this is the video that I showed you early on in the talk. So this is one example of a biohybrid robotic jellyfish swimming in the ocean. You can see that there are lots of fish and natural debris in the ocean. In the background is this rope. You can see, well, the jellyfish is covering it sometimes, but you can see the yellow marker and the red marker, and the electrodes are perfectly embedded, and it's swimming towards the surface, which you can start to see. So it started from the bottom and now it's here. So from videos like that, we can then plot the vertical displacement over time. And when testing this animal and the system out at various swim controller frequencies, we can then compare to our previous laboratory data. So this plot shows the enhancement factor over various swim controller frequencies, and the previous laboratory data for 6 jellyfish are shown here in gray. We tested 2 different animals in the field, And this is the data that we got from them. So we can show that enhancement of in situ or field experiments are actually comparable to laboratory results, where our fastest swimming speeds are about 2.3 times faster than what the animals would naturally do in the absence of external stimulation. But of course, there is quite a lot of variability. And so at 0.75 hertz, you can see that the two animals here perform very differently. And that can be caused by a variety of different things. And we can talk about that if you're more interested later on. But there's just a lot of differences in the animal's own natural pulse rates. And using that hydrodynamic model that I discussed before, we can then also plot the theoretical vertical swimming speeds over the experimental vertical swimming speeds. And you can see that this line of unity is in this black dashed line, and our data actually falls very close to this diagonal line. So our theoretical model accurately predicts swimming speeds with a mean error of 0.8 centimeters per second, which means that using a series of geometric inputs about the animal, we can then predict how fast the animal will be able to swim in the ocean. And so again, for robotic control, we could use all of this to choose which individual jellyfish to deploy in the ocean. So to summarize what I've spoken about today, I hope that I've convinced you that user control of jellyfish locomotion allows the systematic testing of behaviors that otherwise we would just be limited to what the animals naturally do. I've built this biohybrid robot using both a robotic system and a live jellyfish. And this can have advances in terms of biology. So we're showing enhanced swimming behaviors to the animals, as well as robotic advances. So this system is low power and uses between 10 to 1,000 times less external power per mass than existing robots. This is also a proof of concept that we can deploy biohybrid robotic jellyfish reliably well in field environments in which there's a lot of flow and other animals in the ocean. So going back to the spectrum of bioinspired jellyfish constructs, we can then look at robotic constructions on the left, more biological constructions on the right, and our biohybrid robot existing here in this center. So in terms of future work, I want to talk about improving robotic controllability so that we can better monitor the ocean. And so there are a couple of different things that I want to discuss. And the first is that all of our previous experiments were done with unidirectional swing, but of course, natural jellyfish also have more complicated behaviors. So we want to incorporate more turning behaviors as well as closed loop feedback. So by placing sensors on the jellyfish, we want to know if we can have some feedback and be able to basically control a jellyfish through a robotic maze. And so this video shows a series of turning experiments that I conducted in Woods Hole, in which we had one electrode embedded. You can see sometimes you can see this red light flashing, and this chrysiora or sea nettle is actually turning according to this stimulation. So we really want to test various species of jellyfish as well as different turning behaviors. We also want to incorporate these various sensors for closed loop feedback, as well as just taking more information about the ocean. So I've listed a couple of examples here, including temperature, taking sample collections, so various water, as well as GPS data and wireless communication. And really to improve the robotic design, the next phase is to use more biodegradable electronics. So we don't need to worry about what happens when there is debris left over from our biohybrid robots. All of our previous experiments were done with scientific scuba divers carefully monitoring the animal and the robotic system to make sure that we didn't leave any more debris into the ocean. We'd also like to incorporate the animal's own metabolism or even the power of the sea to create seawater batteries to power the 10 milliwatt electronics that we're using. So in order to create a more self-sustaining biohybrid robotic jellyfish. And of course, more broadly, I'm interested in a variety of different bioinspired swimming robots. So this is a figure from a perspective paper that I wrote. You can see various unpiloted underwater vehicles on the left, as well as bioinspired swimming robots. So I'm interested in creating this menagerie of bioinspired swimming robots that we can then use in harmony in the ocean with existing UUVs. And currently, So currently I'm working as an NRC postdoc at NRL, and my project is on using shark skin inspired surfaces for turbulent drag reduction. I don't have much to talk about today regarding that project, but I just wanted to show you a couple of images. So on the left is natural shark skin, and you can see these individual structures called denticles, which look kind of like teeth. And on the right are a couple of images. So this is a CAD file as well as a real 3D printed file of me testing a few different denticle designs out. And if you want to see more information, you can go to this QR code to learn more about what I'm currently working on. And really the last thing that I wanted to talk about before I leave today is I am curating a methods collection in the Journal of Visualized Experiments. And this methods collection is on Bioinspired and Biohybrid robots. So what better place than to come to this seminar and talk to you guys about this. So I've listed this information here. You can go to this QR code to visit the website. And basically, if you're interested in submitting, we'd love to have your abstract submitted by the end of October, if possible. And it's just a 300-word abstract. If your abstract is then accepted, we will invite you to submit a written manuscript, which includes a text of the methods, as well as images. It will go through peer review. And if accepted, then Jove will help script, film, and produce a short 7 to 12-minute video. But you also have the option of producing the video yourselves if you'd like to. So please visit this website and you can click Submit Abstract. So with that, I'd like to acknowledge NRL for hiring me during a pandemic, as well as the members of the Debiri Lab for all of this work that I conducted in my PhD, Cabrillo Marine Aquarium for providing us with jellyfish, our collaborators at the Marine Biological Lab, our scientific illustrators, Rebecca Conta, as well as my funding sources. So with that, I'd like to open up the floor for questions. Thank you.
Great, thank you, Nicole. This is a great talk. I learned a lot about like jellyfish, like tissues and their swim mechanisms. So let's open the question to the audience. So I saw Tuhi already raised his hands. So maybe it's first start from Jimoni. Jimoni, can you ask a question?
Hi, thank you very much for this amazing talk. Yeah, I just put my question in the chat, but yeah, I can ask you speaking. Yeah, it's quite impressive that, I mean, I assume that usually nature does it better. I mean, usually nature performs better. So how could you explain the fact that indeed that jellyfish does not naturally perform better? It's like something like doping for sportsmen, so things like that.
Yeah, so nature tends to be better at many things, but nature is also constrained by a lot of things. So in terms of jellyfish, their evolutionary fitness includes swimming fast, but also includes things like reproduction and feeding behaviors. And so I think their swimming is optimized for a variety of combinations for all of those things and not necessarily just swimming speeds. Although I think they can certainly swim in faster bursts to avoid predators, for example. But I also kind of like to think of this in terms of, I as a human can run faster to places, but there's no need for me to constantly be running around like a crazy person. So instead,
I have no energy principle. So they can do that, but they don't want to do that.
Exactly. So I think there's not much reason for them to do that. But clearly they have the capability of doing that and just need to be prodded by an external microelectronics system. Yeah, thank you for the question.
Disha, do you have a question?
I would like first say, like Nicole, that's a very amazing talk. I'm mainly a fluid mechanics, but learn a lot on the biology side. So to begin with, I want to make a comment with regarding the advertisement you have. I think you should, if you would like to, you can send Chang or me a more formal email, and we can send to the IBiM mailing list, which right now created have 1,069 people registered. So so if you would like to, so
thank you. I will definitely do that.
Yeah, that's like, so my second question, not my first question is mainly about when you actually embedded the system. It seems very simple, but it's still like intrusive, like in a way. How did you hold the jellyfish? Like, what's the process you did that? Because for me, they seem to be very delicate and like I mean, if I'm putting with my wobbly hand, probably gonna break the jellyfish. So can you describe a little bit like your experience like for that?
Yeah, it took a lot of trial and error and practice before I figured out the best techniques. So jellyfish are very delicate, but they are surprisingly more, I guess, massive when you actually reach down and touch them. So they look very delicate in the water, but when you poke them, they are quite fleshy. It kind of feels like a very stiff Jello, I think. So you can imagine that it's difficult to embed the swim controller in the center of the animal as well as the electrodes. But actually, if you hold, so imagine there's a jellyfish in my hand like this, and I'm doing this all underwater. So you would embed that wooden pin into the center of the animal. And because the wooden pin has microscopic barbs naturally, it holds into the tissue actually very well. So we don't need to worry about the center of the animal or the entire microelectronic system slipping out of the animal necessarily. The electrodes are a little trickier. So I had some initial designs where we had like two very straight electrodes, two very straight wires for the electrodes, but then we ended up curving them like a fish hook. And then it's this process of like scooping and making sure that it embeds into the side of the animal. So because the margin has a lot of motion, as you can see in this video, you really need to do it very quickly. And if it falls out, then you move to a different location because you don't want to harm the animal in one place and cause a hole in the tissue. But actually the really nice thing about these jellyfish is that in addition to not having pain receptors and not being more complicated in terms of the ethics, they heal very quickly. So even if they have some massive injuries over the course of a couple of days, they can recover from something like if a chunk of their tissue is missing, for example, like with those tissue stains. So that in itself would only take a couple of days. It might take a couple of hours really for a small hole to close up. So it's just a complicated process of trial and error, but it is relatively simple once you figure it out. So that's the trick, right? You got to make sure that it looks easy to everyone else.
Yeah, it seems if this in the future, if this really take off, like automation, like to a mass production is also like a necessary thing. Like seems to be like a very easy, not seems, it seems to be very easy like process and like repetitive process. Okay, thanks, Nicole. I will give the floor to other people for their questions.
Yeah, thank you.
Yeah, I saw Xinzhang raise his hand. Can you ask your question?
Yes, this is Xinzhang from Chinese Academy of Sciences. I have one question regarding the efficiency. And as you said, the hybrid swimmer can swim faster, 3 times faster than the natural swimmer. And the COT also rises to 2 times the natural swimmer. And you said this seems that the hybrid swimmer is more efficient. I'm just a little bit confused. Since the COT is a metric for quantify the efficiency, if it is 2 times higher than the natural swimmer, it seems that the hybrid swimmer is less efficient. So could you please explain more clearly to us?
Yeah, that's a good point. So in terms of the absolute numbers, the cost of transport would increase. But when I showed that plot comparing our models, our 2 different models, it looked like our cost of transport values were going to be even higher than the experimental data suggested. So we would expect the cost of transports for that higher swimming speeds to be even higher than what we actually got. So in terms of how much speed we're getting versus the increase in cost of transport, it seems like, oh yeah, sorry, you're raising your hand again. Did you have another comment on this?
No. I simply just for the different, yeah.
Okay, yeah. So, you're right in terms of the absolute numbers, but just in terms of what we'd expect at higher swimming speeds.
Oh, okay. Thank you.
Yeah, thank you.
Yeah, I saw another question come from the chat channel. So from Bei Chen, he or she said, asked it seems like, will your device kill the jellyfish or make them die earlier somehow? Did you test their lifespans?
Mm-hmm. No, that's a really good question. That's something that we've thought about. The swim controller is embedded into the animal, but we haven't done any longer term experiments. So we've done a couple of experiments in which we've just had the electrodes embedded and tested to see how they would fare in these oxygen deprived environments. So those were for the metabolism experiments. And we actually ran some of those overnight and the animals were completely fine. When jellyfish get hungry, they tend to just shrink overall. And so, that's what happens if they eat more, they tend to just increase in size. And if they don't eat as much, then they shrink in size. But it would take a lot in order to kill these animals just using this microelectronic system. So in the future, we do want to test, whether this has longer term effects on the animals, but, we're only running them for a couple of hours at a time and it's not affecting them. And once we remove the swim controller, the animals tend to return back to their normal behaviors in terms of pulsing, eating, reproduction. We got lots of baby jellyfish, even while having all of these tests done on them. So it doesn't seem to affect them in the short term. And we do need to do more experiments for longer term.
So actually, I have a follow-up question about this one. So when you said like running for a few hours, I'm wondering, so if for a human, I will simulate like running for a few hours, I definitely will feel exhausted. So for a tiny jellyfish, whether you have noticed that jellyfish might like have less stimulated motion, amplitude motions after like maybe few hours running, or maybe you find a kind of a threshold timing, like you cannot like running longer than this time because jellyfish you're exhausted.
Yeah, that's a really good point. I don't have a lot of quantitative data on that, of course, but just by anecdotal data, it seems like if we tested the animals for an hour, 2 hours, 3 hours, they tended to be okay, like return completely to normal behaviors immediately afterwards. But if we did do tests overnight, then they would be more sluggish for at least a couple of hours afterwards. Like they might just float around like this and occasionally do a weak pulse. But after feeding, after a couple of hours, they would be totally back to normal again.
Okay, that sounds great. So I wonder another follow-up question. So sorry, I have so many questions. So I wonder, so is it possible to turn into such a like a hybrid jellyfish into like a long existing like ocean explorer? Like for example, I have like a flow sensor or maybe other like sensors embedding the jellyfish so we can do some like almost permanent like ocean monitoring. So for example, because it's a kind of simple device that we can duplicate it into an array of jellyfish. So we can maybe build kind of cyber physical ocean sensing network. So maybe in the future, do you think it's possible?
Yeah, I definitely think it's possible. I think we do need to do a lot of work beforehand just to make sure that we are using more biodegradable electronics and they won't harm other animals in case whales or sea turtles end up ingesting these biohybrid jellyfish. But I really envision this array of jellyfish in collaboration with a lot of existing UUVs. So because as I spoke about in the beginning, every model organism and UUVs have advantages and disadvantages. So because jellyfish are relatively slow swimmers, even though we can get them to increase their swimming speeds 3 times, it's still 6 centimeters per second at a relative maximum for these size jellyfish. So if we have a UUV kind of bring a whole horde of these biohybrid robotic jellyfish to a certain area of interest and then release them, they can all travel to different areas of that specific location, take data, and then go back to home base, that UUV, and we can bring them back to shore and look at the data that they collected. So that's one way I think we could deploy these jellyfish in the next 10 to 15 years. But I do think it is really interesting, and I do think that we need a variety of different techniques in order to expand our ocean monitoring capabilities. So
yeah, that sounds very like attracting and fascinating, like future vision. Okay, so is any audience have any questions? Okay, sounds we don't have any question because this is really like a clear presentation. I really like the styling of your presentation, like storytelling, but very clear. This is that exact presentation I really want to learn, to hear, to learn, and to make in the future. Yeah.
Yeah, the QR code is like amazing.
Right, I found the QR code on the right. That you can scan it and see the paper here.
That's the most futuristic thing, man. QR codes.
Maybe should highlight maybe make a marker here says this is the type QR code for the paper or QR code for the video. Otherwise, people might think this is just same QR code, and we don't know what's going on here. Yeah. Okay, so great. So thank you for your talk, and thank you for everyone for attending today's presentation. And that's all for today. See you next week. Bye.
Yeah, thank you so much. Bye.